Welcome to the webinar, Unlocking the Future of Liquid Biopsy Research. Today's presenter will be Dr. Courtney Noah, Vice President of Scientific Affairs at BioIVT, and your moderator will be Dr. Kathy Miller, Senior Director of Product Marketing at BioIVT.
Thank you, Michael. Hello, everyone. I hope you are enjoying your day, and thank you for joining us for this insightful webinar. I'm Dr. Kathy Miller, Senior Director of Product Marketing for Personalized Medicine at BioIVT, and I'm your host today. I'd like to introduce Dr. Courtney Noah, Vice President of Scientific Affairs at BioIVT. She leads a team that provides solutions for BioIVT's clients and business partners. With a broad knowledge of life science applications and a keen understanding of scientific development, Dr. Noah has successfully expanded BioIVT's portfolio and increased our brand awareness. Previously, she served as BioIVT's Head of Marketing and Director of Research and Development for the Cell and Gene Therapy Business Unit. She was also a Senior Marketing Manager for Enzo Life Science and a Senior Scientist at Pall Corporation. Dr. Noah received her PhD in Molecular and Cellular Biology from Stony Brook University and her BS in Food Science from Cornell University. Dr. Noah, the floor is yours.
Thank you very much, Kathy. I appreciate that introduction. And welcome, everyone. Thank you for attending the webinar today. I'm really excited to be speaking on this topic. So today I'll be sharing a presentation on unlocking the future of liquid biopsy research. And what I hope you'll take away from this presentation is really a better understanding of liquid biopsies comprehensively, specifically the applications that they're being used for, and of course, some of the challenges that researchers in this space are facing as they develop assays and trying to make them more accurate, reproducible, and of course, relevant to the patient populations that need them most, whether that be defined by age, gender, or race. So here's the agenda that we'll be following today. And I welcome any questions. We hope to have an engaging discussion as we go through these topics. So before we get into the details around liquid biopsy, I'd like to just take a few minutes to provide some background on BioIVT. As a biospecimen provider, we work with many liquid biopsy clients throughout the space and really help to address some of those challenges that I was mentioning before. We're a global organization. We have over 27 locations throughout the US, UK, and EU. And what you'll see here is the overview of our portfolio, which you can see includes collections from both normal donors as well as clinical collections from patients that have a wide variety of different disease indications. We also have normal human and animal biofluid and tissue collections, and these are often used as control matrices in a variety of different applications. We also collect specimens that are used in support of cell and gene therapy as starting materials. And beyond specimens, we also have a portfolio of products and services that are specifically dedicated to ADME tox research, and these are used in vitro safety assessment studies. So as our clients know, working with BioIVT really brings with it the benefit of our many years of experience in ensuring that specimens are collected ethically and, more importantly, compliant with the ever-changing privacy requirements and regulatory requirements. We're accredited by a number of external organizations, and those include organizations such as CAP, ISO, and the HTA. But what are these accreditations really mean for our customers? Well, they mean a few things. First of all, they include confidence for clinical applications. So specimens collected from a CAP accredited repository have to adhere to formal CLIA approved requirements, and they bring with it specimen collection integrity. You also get the confidence in the pre-analytic variable tracking and control, and that allows samples to be used in drug trials that are associated with biomarker development. And finally, we have an independent audit process that is conducted every 2 years on our clinical sites. And through that, that's really brings with it the diligence and oversight in making sure that our clinical collections are performed according to the protocols that we have specified and that our clients expect. So as we look a bit further at our specimen offering, I'd like to provide some more detail on the breadth of our offering. And you can see here the variety of different disease indications. We also collect across a number of different matrices and formats. We actually work with over 450 different global sites to collect biofluid and tissue specimens. And most importantly, each collection is conducted according to one of our 40 IRB approved protocols. And that really provides a lot of different options for our clients in terms of the specimen type that we're collecting or the format that they receive that specimen in. But the specimen is actually, as we know, much more than just that tangible product that you receive. It's all of that associated data that comes with it. And that can include the details around the donor, their demographics. It could include the details around their medical history, but it can also include characteristics of the specimen itself. And those can include details such as the mutation status, the genotype, or next-gen sequencing data. So now hopefully you have a better understanding of BioIVT. So let's shift over into the main topic for this presentation. So I assume many of you are here to learn about liquid biopsies and, like I mentioned, the future potential and how they can impact patient care. And while we've seen a lot of great advancements, especially over the past few years in this field, we've seen the approval of recent assays such as the plasma focus assay by LabCorp, additional assays coming forward from Garden and Foundation Medicine. We know that more still needs to be done in this field to really ensure that the next generation of liquid biopsy assays provide the most reliable results and that they're really personalized to the donor populations that are most affected by these diseases. So let's start by just that overview of liquid biopsies. And when you look at the term, what first comes to mind is, of course, it's a blood test. And that is true, but we'll talk a little bit that liquid biopsies can be beyond blood. It's really a biofluid sample. And the other part of that word biopsy, it seems to imply that a liquid biopsy is replacing the need for a tissue biopsy. But I want to call out here that that's a bit of a misnomer. Liquid biopsies have yet to really entirely replace that competition of the solid tissue biopsy. Further to that, developments in the space go beyond just the potential to replace tissue biopsies. In fact, they're actually looking at replacing other diagnostics and monitoring tests that are in existence today. One example of that is if we look at patients that experience recurrent disease from the persistence of residual tumor cells. This is what's called minimal residual disease, or MRD. MRD can actually not be identified by standard radiological exams or clinical evaluation. And so this is where there's an opportunity to look at liquid biopsy assays and the potential application that they have in those types of situations. The analytes that researchers are exploring in terms of liquid biopsies can include anything from circulating tumor DNA or ctDNA, circulating tumor cells, CTCs, extracellular vesicles, and circulating RNA. And these biomarkers provide a variety of insights. They can provide information pertaining to disease presence, prognosis, treatment response, and as I just mentioned before, minimal residual disease detection. And as you can see here, we've called out some of the advantages to liquid biopsies. There are many, which is why it's an actively growing area. Most important for the patient is that they're less invasive, dangerous, and painful. They also provide a more comprehensive molecular overview of tumor heterogeneity. And this can possibly reflect some of the characteristics of different cancer locations in a particular patient. So what's important to note here is this can't be achieved by a single tumor biopsy. So it really is an advantage in that situation to use a liquid biopsy. And finally, another benefit is it is much easier to obtain repeated blood samples over time to understand the dynamics of response to a specific treatment. But of course, the flip side of this is that there are some disadvantages. There are aspects to liquid biopsies that really limit their potential, and these are related to sensitivity and clinical validity. And we'll go into these in more detail in the next slide. So when researchers are working on developing any diagnostic assay, but as we're talking about today, liquid biopsy assays, they're working on developing a high quality, high value assay. And so the questions that they're asking are, how accurate is the test that I'm looking to develop? And how does this improve overall patient outcomes? So pertaining to accuracy, this is where pre-analytical variables can have a great influence on the results. So it's super important to understand the factors that influence the analyte of interest. So if we look at ctDNA analysis, really making sure that those pre-analytical variables are controlled and optimized for that type of analysis. Today, there still remains that discordance between solid tumor profiling and liquid biopsies. And so these liquid biopsies have yet to be used as standalone assays. But the further we can define that limit of detection specific to different genes across a panel, the more we'll work to improve that analytical validity of the test. And there continues to be an emerging evidence in support of the clinical validity of liquid biopsy. Certainly more work needs to be done to improve applications directed at quantifying tumor origin and minimal residual disease, but there's definitely great promise in the future clinical utility. And as we've already seen, there's been successes in the space in terms of these assays being used to determine targeted therapies that will be most relevant based on the detection of genomic markers. So let's dig in on what future optimizations are required to help improve that clinical accuracy, reproducibility, and of course the patient impact. So as a biospecimen provider, we can't overlook the importance of that critical first step in controlling the sample. So let's start there. When we look at that pre-analytic phase, it can really be divided into two main segments. The first is that pertaining to the patient. And as you can see, the factors here really can't be controlled. They are what they are. They're associated with the patient. If we look on the specimen side prior to that analytic phase, this is where specification and control are much more feasible. To date, there's no comprehensive set of recommendations regarding biospecimens and methods for evaluating pre-analytical phases of a test. And that's really because the factors that determine that optimal processing of a patient sample are influenced by the specimen being collected itself, the analyte, and the assay. So we do have to consider those as we look to see what pre-analytical factors or considerations we have in those collections. New liquid biopsy assays are often targeted at looking at DNA, RNA, and proteins, and they are trying to do this in a much more quantitative and multiplex manner. So with this, there's become a much greater appreciation of the importance and the fragility of biospecimens and biofluids in particular, often with regards to their molecular targets. The collection of biospecimens through analyte optimized protocols will certainly enhance the diversity and the quality of the end assays that researchers are looking to develop. So when we look at the industry, certainly there's much awareness around the gaps that we have in our standardization practices. And academic, industry, and government groups have started to come together. And you can see examples of some of the groups in this space that are really trying to put out guidance, put out recommendations, and also partner together to kind of put the information that they know to best use. So they've come together to define standards and improve the clinical utility, really driving forward precision medicine. And while I don't have the time in this webinar to go through all of the recommendations that they've put out, I really encourage you to seek out more information from these organizations on their website. They've gathered a lot of great information over the past 2 to 3 years to improve the use of liquid biopsies in oncology practice. But just to call out one example here, I did want to share some information that's been put out by BloodPAC, which is a consortium with a mandate to accelerate the development and validation of liquid biopsy assays. They have a working group, their pre-analytical variables working group, that identified and created 11 minimal technical data elements, looking across all different critical variables. And they've required this for any new studies that are submitted into their data commons. And this is really important because what this brings forward is an enhanced ability to compare data sets across different studies, allowing you to draw more conclusions on the impact of specific pre-analytical variables. But also, it presents a common vocabulary, and I think that's really important to call out because as we're speaking different methods and different variables and controls, it's important to be using a common vocabulary so that we're consistent and appropriate with the meanings that we assign to things. So I won't spend too long on this next slide, but I did want to show how BioIVT has worked to standardize our collection processes. And so if you look here on the left, you can see an example of our double-spun plasma protocol. We collect this actually as a matched matrix type with many of our tumor tissue collections. And just like the guidelines that were provided in the prior slide, the data points are noted for the collection tube that we use, the processing time, storage temperature, et cetera. However, as I previously noted, there is still that need for flexibility. So depending on the client that we're working with or the assay that they're working to develop and its specific requirements, we do work with our clients to adapt collection protocols to make sure that they are fit for purpose, but still maintain that consistency and the control of the collections. So ultimately, success in translating and translational research for liquid biopsies will require standardization of the specimen handling and processing along the lines of what's currently seen in the field of clinical chemistry. I do believe that this will lead to improved biomarker testing and performance. So now we're going to kind of switch topics a little bit, still related to liquid biopsies, but this is an area that has definitely gained a lot of interest in the regulatory space, as well as across biotech and pharma, and that's the topic of diversity and making sure that we're considering diversity in the plans that we put together for clinical trials. So most recently in December 2022, there's actually a law signed by President Joe Biden that requires researchers and companies seeking approval for late-stage clinical trials to submit a plan for ensuring diversity among trial participants to the FDA. And what this does is it really highlights the need and the urgency to overcome some of the barriers that we see today in including appropriate patient groups and making sure that they're participating in studies. So when addressing disparities, as well as those specific to cancer, it's really important to acknowledge where we're at today. And so I have a few examples that I've collected here, really to try to show some of the evidence and some of the data that's collected across looking at different disparities. And it's not just pertaining to disparities related to race and ethnicity. We also see that pertaining to age and gender. And although clearly this has become an important policy priority, what we see is some of the progress has really been stalled on getting that participation of minority population groups. That said, we still need to make inroads, and particularly as liquid biopsies are starting to come more online, gain more traction, we need to make sure that they are being designed with equitable clinical research programs that match the demographics of the disease. So this is really where I see an open door. And if we think about where can we start and where can we gain traction, if we look at the preclinical space, which is where BioIVT is providing samples to our clients for their research purposes, I think there's a lot of opportunities to gain insights into demographic-driven factors. So this brings me to speak a little bit about BioIVT and our donor centers. We actually independently operate 10 donor centers throughout the US and the UK. And as you can see here, we have over 10,000 unique donors that we see every year passing through those centers. And they collect a variety of different matrices that can include blood and bone marrow, non-blood biofluids, leukopaks, and bulk plasma. And I think accessing this donor cohort is really important as we think about liquid biopsies, because this presents a unique opportunity to assess those healthy normal populations for assay sensitivity and specificity applications and uses. And these capabilities have been used by many of our liquid biopsy clients in support of their validation studies. And as you can see here, when I was talking about making sure that we're looking across different age groups, across different genders, and across different races or ethnicities, we do have a broad donor base that spans many of those different areas. But also, it's important to call out, we have our own recruitment team. And so we're many times working with clients who have unique needs, are looking for very specific risk match controls. And we can work with them through the work of our recruitment team to engage and work with donors in the area and bring them in for collections. In addition, if we look on our clinical side, we've actually done an assessment here. What you can see on the right-hand side is looking at the breakdown across gender, age, and race of our lifetime clinical collections, and compared that to what we see in the US population. This is based on the most recent census data. And we were really pleased and happy to see the spread and how that compares against what is seen in the typical US population. So while we do have a skew towards Caucasian white subset, there is good representation across the other racial groups. And as we start to look at gender, there's actually more participation with females, and even in the older age group, there's more participation there, which is important to call out because these are oftentimes groups that are underrepresented in clinical trials. So here's where I'm calling out, utilizing these specimens during early liquid biopsy development and discovery can help to provide better insights to inform and support those diversity plans that you'll be putting into practice in the late stage clinical trials. And as we know, as you're putting together diversity plans, you have to think of what challenges am I going to encounter in terms of recruitment. And there's many. I listed a few here, and I think top of mind oftentimes is mistrust. We know that there's been some historical unethical practices that have led to underlying anxiety. I think the most notable example of that is the Henrietta Lacks case. But interestingly, if you ask individuals, and there has been research around this, many individuals from ethnic groups say that they are eager to participate. So there's other factors in there that's preventing them from enrolling and participating. And so those lead down to the other items listed here. So it could be just general lack of comfort with the process. Perhaps there's been underlying fear or maybe opinions of other family members that have led them to not want to participate. It could be lack of information about the clinical trial that's really relevant to that audience. I think one of the big things can definitely be time and resource constraints. So we certainly know there's a lot of logistics around participation. How am I going to get there? What time is it going to take? Is there some type of financial burden that participants will encounter? And finally, just a general lack of awareness. And I think many groups have started to come together most recently and really making sure that there is more information out there and that it's presented in a way that's relevant to their target demographic. But this is really where we want to put out a message that BioIVT is very eager to partner with institutions, practices, advocacy groups that are engaged in these efforts to expand access to clinical trials. We want to do our part in really supporting this through that preclinical research and engagement with patients and donors. And we hope that through this, we can really help to accelerate the advances in the field of liquid biopsies and drug development, certainly as a whole. So I want to just take a few minutes now to drive home the message behind the importance of preclinical research. So as it pertains to liquid biopsies, if we think about the population, it's diversifying organically, and there's a greater need and urgency to support the generalization of findings. So with the collection of more enhanced data during those preclinical phases of research, there can be a more informed development of the clinical guidelines in the later phases. Secondly, if we look at the genetic, proteomic, and epigenetic profiles of different populations, whether that be across age, race, and gender, these can definitely vary significantly. So without that early stage assessment, again, in that preclinical phase, using those specimens collected through organizations such as BioIVT, you can start to identify more unique targets. If we don't look early on, those targets can go unnoticed, undiscovered, and really minimize the potential opportunities for precision medicine. And finally, as I mentioned about the relevance of information provided to clinical trial participants or audiences that you're trying to recruit to participate, compiling representative data can help in gaining that trust and educating patients that are most critical for clinical trial enrollment. So the more evidence that can be gathered in that preclinical phase that's relevant, that's applicable, can really, I believe, have a tremendous impact and highlight the value of participation to those individuals. So as I spoke about today, there's a need for standardized, controlled collection procedures derived from a diverse patient population to really best ensure that we're creating and developing effective and impactful liquid biopsies. And so there are many biospecimen providers in the space, but I really challenge you to carefully evaluate them against these critical elements. First being experience. How many clients have they worked with? Have they gone through that process of really designing those fit for purpose collections? What are their quality control and standardization procedures look like? How diverse is their donor base, whether that be through donor centers or clinical collection sites? And ultimately, what's their commitment to driving smarter science and supporting precision medicine? And so this is really baked into the BioIVT mission statement, but I have to say the proof is in the publications and the utilization of the clients who seek out our donor and clinical specimen collections. And for those of you who may be interested, please reach out. I'm happy to share publications of some of our liquid biopsy clients who have published using our samples, and you can see examples of how they use them for their validation studies. But I see lots of opportunity ahead for liquid biopsies, whether that's in the broadening and applicability of their use, increasing the reliability, but certainly most important for me is really improving the access and the availability for patients who can benefit most. So with that, I want to thank you very much for your time today. I think we're going to open it up for questions at this time. And of course, if you have any questions that come to mind after this webinar, you have my contact information. Please feel free to reach out to me directly.
Thank you, Courtney. That was very interesting. I was happy to actually be able to attend it this time. I do have a few questions that have been submitted, but if there's any additional questions, please do enter them into the questions window of GoToWebinar, and we will make sure that we get them answered. The first question I have is, as this is a newly emerging diagnostic area, are there any guidelines that can help liquid biopsy developers understand the requirements for clinical validation?
Yeah, so like I said, there's lots of information out there, and I think the organization that I would point you to is to look at BloodPAC. Again, they have many great resources, and I think about 2 years ago, they put out a special report in clinical chemistry that provided some guidance around standard methods and protocols. Within that, and BloodPAC is actually an organization that has representatives across industry, I believe some academia, but certainly those regulatory groups as well, including the FDA. So with that, they kind of called out the recommendations on the sample types to be used, the experimental design, statistical analysis, and even data presentation. So I think it's a really good guideline and guidance document for anyone in this space. And I think for us, as we're thinking about it, one of the interesting things that they called out was around normal controls, and really recommending that in use of normal controls that you're using individual samples rather over pooled, and that those be age and risk matched. And that's where I think there's great opportunity for anyone looking for those normal controls to utilize the services and capabilities at our donor centers, which can really quickly recruit and consent donors that are specific to the liquid biopsy developers' needs.
Very good. Another question I have is, can BioIVT collect controls for liquid biopsy assay development in a clinical setting?
Yes, so I was just talking about the controls that we can collect at our donor centers, but some of our clients are interested in normal control samples, so healthy patients that are still within the clinical setting. And actually, that's a very common request that we get, and we can collect normal samples in a clinical setting. And so that comes with the additional data that you would get, including medical history and anything else that you would expect from a clinical setting collection.
Excellent. How can I ensure that the specimens I'm using in my validation studies will be acceptable to regulators?
Yeah, so that's a great question. I kind of touched on it a little bit in my presentation, but there's so much more we can say on this topic. And I would say kind of first and foremost, never assume that a sample you're using has been collected in a compliant process. That's why it's very important that you're asking questions before you build your test cohorts of whoever you're working with, even patient advocacy groups and things like that, because it's so important to have everything locked down, controlled, compliant early on. So at BioIVT, we have various forms of informed consent that are utilized for the collection of human biospecimens in different disease areas. These forms undergo a review process according to regulatory and ethical authorities, including groups such as IRBs. And for anyone who would like more information, because there's really, like I said, a lot we could talk about related to this question, this topic, BioIVT has a great amount of information on our website. We have webinars that we've done and some white papers on questions to ask to really make sure that you're comfortable and compliant in the samples that you're using.
Good. I have a few more questions. We're in early stages of our discovery and want to use matched tissues to biofluids. Can BioIVT feasibly do those collections?
Yes, so many of the samples, I can't say all of them, but in many cases, we're actually collecting across a number of different matrices types when we collect from diseased individuals. And so that will include the tumor tissue itself, along with biofluid collections. And as we've seen, liquid biopsy discovery and development gain traction, we're oftentimes collecting the tumor samples with that double spun matched plasma as well. So the two can go together as a match set for those development needs.
Good. Do you see liquid biopsy applications expanding beyond the use in oncology indications?
Yeah, so some of the conferences that I've been attending, certainly there's a large focus on oncology, but I see kind of where there's starting to be an uptick of interest and gaining some traction is in the application of liquid biopsies for CNS applications. And I think there's a lot of lessons that can be learned through the work that's been done in looking at liquid biopsies for cancer applications. So hopefully CNS has a leg up and can kind of use that knowledge as they start to look at whether it's blood or CSF samples for detection of biomarkers. And that's a good call out too, as we're talking about CSF and CNS related specimens. BioIVT actually just recently acquired Precision Med, which is one of the premier biospecimen providers of CSF with related match samples of blood collection and extensive patient data. So for those who may be seeking those types of samples, definitely inquire with us. We have a really broad collection, and also that includes some longitudinal samples too, which can be really valuable as we're trying to kind of track the outcomes of different patients.
Yeah, I'm excited to look more into the longitudinal samples myself. Do you have molecular data about the samples, such as specific mutations?
Yes, so that's something actually that we've been expanding, I don't know, Kathy, past like 3 to 4 years here at BioIVT. That's definitely a need that we've seen from our clients, having more genomic data that they can use in their discovery development processes. So we screen about 800 plus samples using next-gen sequencing for across a number of different oncology panels. And so those are accessible, and depending on the mutation of interest, please reach out to us and we're happy to collect an inventory report that meets those needs.
And as a follow-up to that, we have, do you have a catalog of the mutations that we have screened for? So.
Yes, we do have a report of the various panels that we run. You know, that said, similar to what I was saying before about fit for purpose, we have our standard, but we also work with clients who may have more unique needs, and those can be discussed on kind of a custom screening program.
And that is all the questions I've received so far today. So
great. Well, like I said, if anyone has any other questions that come to mind, my contact information is in this presentation, so please don't hesitate to reach out. I'm happy to answer questions following this.